Okay, so this is going to be chapter 3. So we're going to be talking about cell structure and cell function. So first of all, we're going to take a look at what a cell is, what the components of a cell is, what their function is. We want to start off first of all with the cell theory. So the cell theory is basically what governs kind of all of our cell structure studies. And let me just double check on one thing. And so essentially what we have in the cell theory is essentially the idea of sort of a theory that is designed to describe the nature of the cell. And for the most part, that description of the nature of the cell itself starts off with the definition of a cell, which is the basic unit of life. Now, when we say what is the basic unit of life, a lot of times students say, what does that mean? That basically means that this is the smallest that we can get and still answer all the big questions of life, those big biological questions of life. So energy, acquisition, reproduction, growth, development, things like that. The other component of the cell theory is that everything that's living is made up of cells. And so that we know, and reason is because we have never made an observation where a living thing is not made up of cells. And then the last piece of the cell theory is that, answers the question, where do cells come from? And that question is answered by, they come from other cells, just like dogs come from dogs, cats come from cats, cells come from other cells. And so when we take a look at cells, they kind of have a lot of variation in terms of their structure and function and the things they can do. For instance, blood vessels are made up of very specific cells. Red blood cells, for instance, are a very unique type of cell. Nerve cells have a very different look to them as well. Osteocytes, which is a bone cell, kind of hiding in that little dark cavity right there in your bone. That also has another very distinct type of personality to it, but they all have some commonalities to it. They all have the same working parts, basically. And we're going to take a look at those working parts here in this chapter. The first thing we want to do, however, is we want to kind of break down a little bit about the work that the cell has to do. So in order for you to survive and live, your cells have to do certain tasks that have to basically answer all those big biological questions of life so that you can grow large enough and capable of to reproduce. And so when we talk about the work of the cell or what the cell can do, the first thing we're going to be talking about is how much work can it do given its relative size. So we refer to that as what's called the surface area, or SA, to volume ratio. That is to say that the surface area represents basically how big you are. So you can think of the surface area, say the volume of the cell, and of course, excuse me, backwards. I got ahead of myself there. You can think of the surface area as the circumference of the cell. And then, of course, the volume. So literally, you can think of the cell as a sphere, essentially. Now, generally speaking, whenever you take a look at the size versus the surface area is important because this is the working space. This is what you have to work with. This is your working surface. So if you want to do some sort of a task, you have to have a lot of surface area. This is also, we can refer to surface area as kind of like a benefit. That is to say that the more working surface you have, the more you can do. The volume, on the other hand, is kind of like the cost, right? So you basically have to pay the costs of making a larger cell. So generally speaking, and usually if you have a large cell, that means it's a little bit higher cost. So when you take a look at cells and you compare small cells to large cells, smaller cells oftentimes tend to have more surface area. That is to say that you have smaller costs with greater benefit. Now, oftentimes what will happen is in order to sort of reduce costs and increase benefit, cells will manipulate themselves so that they can increase their surface area, but not their volume. And so there's a lot of different tricks associated with that. And so, like, for instance, if you take a rectangular cell like this, and let's say that's your working surface right there, just that little top part, you can only get a little bit done with the size of that cell. So what if you wanted to increase your surface area or your working surface? Well, one of the things the cell will do is it'll basically take that surface and it'll kind of start to create all these like little kind of protrusions on it. Now, all of a sudden, what you have is not just that one surface, but now you have all of this surface that you can work on. So what you've done is you've effectively increased your surface area while maintaining costs. So that's basically the economy of the cell. So here's kind of a good comparative. The basically compares three different systems. In one, we're going to have a 4 centimeter cube. So you have 4 centimeters here by 4 centimeters by 4 centimeters. Okay, so it's a 4 centimeter cube, and you have a certain amount of working space. So all of this area right here would be your working space. And so your total surface area in this particular case would be 96 square centimeters. Now, what happens if we cut this up into not just one cube, but we cut it up into 8, so that are 2. So here is 2 by 2 by 2 by 2. And so in this case, all of this right here is all your working surface area, but you also have this piece right in here that's kind of on these other faces that you didn't have access to before. So you've actually increased your surface area now to 192 centimeters squared. Now, if you basically do cut that up again, so basically cut these guys up as well, then what you're doing is you're increasing your surface area even more. The important thing to remember is notice the volume hasn't changed. Here the volume is 64 cubic centimeters. Here it's 64 cubic centimeters, here it's 64 cubic centimeters. So notice you haven't taken up any extra space. All you've done is cut up what you have to reveal and expose more surfaces that you can do work on. So when you take a look at your surface area as a function of volume, basically in the first system, you have 1.5 to 1 surface area to volume ratio. In the second one, you double that to 3 to 1. And in the third one, you double it again to 6 to 1. So you can see by increasing or exposing new surfaces, you can generate a lot more working surface and productivity without incurring a lot more costs in terms of building a larger volume cell. So now we want to do a little bit of a topical shift. So we want to go to some of the tools of how we study cells themselves. And of course, the main tool that we use to study cells themselves is the microscope. So we're going to be taking a look at some microscopy. Mostly what we use is what's called the compound light microscope. That's the microscope that most of you are probably used to. And so generally what microscopy does is it improves resolution. And so resolution is defined as the ability to distinguish 2 points. Now, this is difficult to do on a screen, but imagine that you were on a whiteboard. If you have 2 points, you could probably see those 2 points as 2 separate points pretty easily. As I moved them together, it gets a little harder. And as I put them like right next to each other, it gets a little harder. And so you're losing resolution. So what microscopy does is it basically gets you an up-close personal look at these so that you can see that these two are still indeed 2 separate spots. Now, when you take a look at all the microscopes and the types of microscopy that we have, the basic generic light microscope is probably the least powerful of all of them. And so essentially what you do is you just use a series of lenses and light, and you essentially magnify, just like a magnifying glass, the image as the light passes from the image to the lenses and makes them look larger. And so that kind of allows us to be able to see the specimens. So when we take a look at resolving power, right, so the ability to differentiate between 2 spots, if you use the naked eye, you don't have any magnification there, but if you use the naked eye, you can basically discern down to about 0.1 millimeter. That's about 100 micrometers. That's 1/10 of a millimeter. And so that's pretty tiny. That's pretty good, actually. A light microscope at 1000x will get you down to a little bit more. So that'll get you down to 0.1 micrometers. If you use an electron microscope at about 100,000 magnification, you can get down to 0.01 micrometers. So you can get a lot more resolution with some of these other microscopic types of techniques. So let's take a look at some of the images that we get here. So in this one right here, we can see this is a light micrograph, basically of a blood vessel. It looks like an artery. So you can see the red blood cells in here, and you can see the arterial wall, this is the blood vessel wall. And so you can see we use pink stains basically to create contrast. because ultimately cells aren't pink. They're clear, they're translucent, they don't have color. So in order to be able to see them with light microscopy, we use stains. And so staining in biology is a very, very common trick. In B, now we have what's called an electron micrograph. So basically, instead of using light, like a light micrograph does, in this particular case, you're going to use electrons instead. And so here you can see it looks like a very kind of a detailed look of a cross-section of a red blood cell of an artery. Still the same thing that we're looking at. And then when you take a look, that's a transmission. That's a very special type called the transmission. So a transmission electron micrograph will basically take your little specimen. It's usually like a cross-section of your specimen, and you'll beam electrons and they'll pass through, and then you'll detect it as it passes through. In a scanning electron micrograph, basically what happens is you have a specimen and then you beam electrons on it, and instead it glances off and you basically analyze the reflection. So it gives you kind of this three-dimensional topographical view of what you're looking at. Light microscopy is similar to scanning in the sense that it takes a photon of light and it basically bounces that off of your image, and that reflection is ultimately what you see as it passes through these lenses that magnify the image itself. And so that's kind of like the three main types of microscopy that we're going to be taking a look at. And so let's kind of zoom in a little bit on the compound light microscope. So ultimately, we will have some images that we can see with the human eye, some that we cannot. So if we cannot, we basically put a bunch of lenses between us and the reflected light. That's going to magnify the object. It's kind of basically the same thing you do when you hold up a magnifying glass. And then that'll allow you to be able to just see it very easy. It's a very easy, very, you don't need a lot of high tech to do this. If you do electron microscopy, you need like vacuum encasement and stuff like that. So it's a little bit more of an ordeal to use the other types of microscopy. Light microscopy is the easiest. Transmission electron microscope will give you a two-dimensional image. So basically use a stream of electrons to magnify the images as it passes through the specimen itself. And the scanning it makes a 3D image, and it'll basically glance those electrons off of the surface of the structure, and thereby giving you greater magnification because electrons are so tiny. Okay. So now let's go ahead and take a look at the organization of the cells themselves. So first of all, we want to take a look at what kinds of cells do we have in nature. So the first thing that we're going to have in nature is basically either prokaryotic cells or eukaryotic cells. Now, if you're a cell in this world, you will fall into one of these two categories. These are the two different types of cells that we see. A prokaryotic cell is one that lacks a nucleus. Okay, so that's one thing. A eukaryote, by comparison, will have a nucleus in it. Also, in a prokaryotic cell, you will have basically two major groups. So we usually associate bacteria as a prokaryotic, another group called eubacteria and archaebacteria, which are all prokaryotic. The eukaryotes include the things that we're probably more familiar with. For instance, animals like us, plants like the things we grow and eat, fungi like mushrooms, which some of us love, and then of course protists, which are like little single-celled critters that are kind of like pond scum. Okay. So those are the two different types of cells. Either you have a nucleus or you don't. So now let's go ahead and take a look at the commonalities of cells. So what are some of the things that all cells have? So first of all cells have to have a plasma membrane. That is to say, they have to have this sort of membrane that sort of bounds them, creates an inside and an outside. So it surrounds the cell. It's the periphery of the cell. And it's made out of phospholipids. Phospholipids basically, and we'll talk more about this later on, but phospholipids are essentially your polar head. We talked a little bit about this in chapter 2, and your non-polar tail. So those are your phospholipids. And then, of course, as they organize, they form this lipid bilayer, which is selectively permeable. It allows some things to come in and does not allow some things to come in. So it kind of picks and chooses who gets to come into the cell or who gets to leave the cell. The other main thing that all cells have to have is cytoplasm, which is essentially the semi-fluid substance that's inside the cell. So now you have a membrane and you've got all this fluid and stuff inside the cell. In addition, it also includes all the organelles. So you'll see like these little bubble-like membrane-bound structures inside the cell. Those are all organelles. These are compartments, if you will, membrane-bound compartments. That do a specific task. Usually what happens is they're answering some sort of one of those big biological questions of life. Either they're working on energy or they're working on structure or something of that nature. So this basically is kind of like a typical eukaryotic cell. So you can see you've got the nucleus here. And you see I have all this chromatin in here. The nucleolus is this little guy right in here. So this guy right here is the nucleus. We're going to see this up close. Let's see if I can do this. Is that better? There's the nucleus. This guy right here is the nucleolus. The chromatin is all this kind of bluish stuff. That's the DNA. You have your endoplasmic reticulum out here. And then, of course, your nuclear envelope, which is bounding your nucleus. So we're going to take a look at that. One of the things you want to be able to do in a chapter like this, that's a pretty common sort of a thing, is taking a look at something like this. And then, of course, a common set of questions would be, what is it and what does it do? So what is this? I'm going to point to a structure, for instance, and what does it do? So if I point to mitochondria, you should be able to say, oh, yeah, that's mitochondria. It's basically the ATP producer of the cell. Okay. So an image like this is a really, really good one to use for your studies because you have all the organelles laid out here in this, which is basically an animal cell, and you have a little bit of detail about their function there. So if you were to memorize this image, you would have a pretty good start to memorizing and understanding the cell structure and the organelles structure and function that we're going to be looking at for the rest, pretty much for the most of the rest of the chapter. So this one, I would say make this one a priority in your studies. So then the other thing that we also want to take a look at, so is the evolution of the eukaryotic cell. So this is what's called the endosymbiosis theory. There's a couple of different versions of this, mostly because it happened some 3.5 billion years ago. So it's really difficult to know for sure how any of that happened because there's been just so much trauma on the planet and so many things that have happened in 3.5 billion years. There's really no direct evidence left at all. And so what we have is just kind of circumstantial and indirect suggestions of what might have happened. But we have pretty much roughly about a half dozen different possible stories of how this could have all come down. So here's one of them. And so the idea was that the first cells that came to be, because they're the simplest, were the prokaryotes, or the archaeans, really is what we think they were. And at this particular point, they were anaerobic. So the Earth had no oxygen. So these organisms were what's called anaerobic. They do their metabolism without oxygen. And so ultimately, these guys were usually extremophiles. They live in very harsh environments. And so perfect for old age Earth. Okay. So what we think is that basically how eukaryotes came along is that, or how they kind of came to be, or the organelles, is that basically an ancient eukaryote essentially engulfed a prokaryote. And that prokaryote kind of became the organelle itself. So here's kind of what it looks like in pictures. So here you can see you have your original prokaryote, which basically has no nucleus. And then there's a couple of different versions of, well, how did the nucleus come along? And there's a couple of versions there. But ultimately, this kind of evolved into a eukaryote. where there's a lot of ideas there. We don't really know exactly how all that happened. But the idea was that what happened was this eukaryote basically engulfed these prokaryotes, just the same way your white blood cells engulf bacteria when they do battle with bacteria. But what happened was one of these prokaryotes actually turned out to be beneficial. Like, for instance, they were making energy for this eukaryotic cell. So it's kind of one of those things where it's like, you're more valuable to me alive than dead, so I'm going to hang on to you. So ultimately, that became the mitochondria. And then the other one that was engulfed was what's called a little cyanobacterium. It's a bacteria that has the ability to do photosynthesis. And so it engulfed it. And so it said, you know what? Hey, you're pretty useful. You're making actually molecules that I can actually make energy from. So you're more important to me alive than if I were to eat you. So they hung on to them, and those basically became chloroplasts. And so you have these two different lineages, one that engulfed a pre-mitochondrion to become animal cells, and one that engulfed a pre-mitochondrion and a photosynthetic bacteria to become plants. And so that's kind of the endosymbiosis theory. That's kind of like I said, there's a lot of narrative, there's a lot of storytelling in that one, because there's not a lot of direct evidence for us to really be able to know for sure. But there's about a half dozen different versions of that story, and we really don't know which one of them is true. It could be something completely altogether that we haven't considered as well. So now let's go ahead and take a look at the plasma membrane. So the plasma membrane basically is a phospholipid bilayer. And so it's a bilayer that has membranes embedded in it. And so ultimately, if you were to put phospholipids, like inject phospholipids into water, what happens is they naturally and spontaneously form the bilayer. So immediately they'll just basically do something like this. They'll kind of arrange themselves to minimize their contact with water, and they'll do something like this, kind of forming a bilayer. Now within that, so what that does, it minimizes energetic tension, because it takes these little polar hydrophilic heads and points them toward water, which they like, and takes these little hydrophobic tails and sort of hides them inside, so they don't have to worry about being touched by water. So it's exactly what they want. So it happens very spontaneously, just the same way the oil stratifies when you put it in water. And so when you start to insert the proteins in it, then ultimately what you end up getting is something that looks like this. So here you can see your lipid bilayer, here, your polar heads, and your hydrophobic tails. Now, in your membrane, you also have an additive. You have cholesterol added in there. That basically stabilizes the membrane from temperature fluctuations. And you have a lot of different proteins, right? So for instance, you have a protein that is basically inserted, like this guy right here, inserted into the membrane. You have some of them that are not inserted in the membrane. They're just kind of hanging out there. They're kind of membrane associated peripheral proteins. You also have some proteins that have these little carbohydrate branches on them like this. So that's called the glycoproteins. You have a protein with a little carbohydrate branch on it. You also have some of these carbohydrate chains that are actually attached to the phospholipids. That's called a glycolipid. So you have all these different things that are embedded in the membrane, and they all have various functions. So it's a very, very multifunctional area in the membrane itself. The model that we came up with in order to describe the nature of the membrane is called the fluid mosaic model. That basically means that proteins, that is to say a mosaic of proteins, fluidly move in a fluid membrane. So they drift around, basically. They kind of move around laterally and so forth. And so that's kind of the model we had. And of course, within this, we have a couple of very important proteins. For instance, we mentioned cholesterol, which stabilizes the protein, or the membrane rather. We have glycoproteins and glycolipids. These guys are important because what these do is these are essentially the cell ID system. So for instance, if you've ever wondered, how does a liver cell know it's a liver cell, not a neural cell? Well, it's because the glycoproteins kind of create a little profile that sort of tells it that it's a liver cell. It's almost kind of like a biochemical barcode. Okay, so it's like if you've got a particular profile of glycolipids and glycoproteins, then that is the pattern for a liver cell. They also happen to be your self tags. So every single cell in your body has basically a self tag. So every cell in my body basically has mark written all over it. And so that's the way your body determines whether or not something belongs in your body or not. So if I have something in my body that doesn't have my self tag on it, mark, then my body attacks it. And that's kind of the basis of tissue rejection. So I get Bob's liver, but the problem is those cells have Bob on it. My immune system wants to attack it because Bob is foreign. And so therefore it has to attack. So some proteins, not just glycoproteins and glycolipids, will act like channels. And so what they do is they basically will create safe passage for molecules that cannot otherwise get through the membrane. So if you have a little molecule here, it'll allow safe passage to go through the membrane. And generally speaking, these are going to be small polar or charged molecules. Generally speaking, small hydrophobic molecules can pass freely through the bilayer. Why? Because the bilayer is nonpolar. So what that means is things like oxygen, which is also nonpolar, or CO2, which is also nonpolar, can freely move through these nonpolar membranes. They have no problem with that. So if you're a water and you're polar, or you're an ion, you're charged, you need a channel. Water itself, being polar, has a special channel called aquaporins. And that's basically, and they're mostly open, and so your water just basically freely moves in and out of cells as it wishes. So let's take a look at some of the membrane functions. So these are some really important functions that we're going to take a look at later. So essentially you have diffusion. I'm not going to expand on these now because the chapters will expand on them. So you have diffusion, osmosis, facilitated diffusion, active transport, and endocytosis and exocytosis. So this is basically transport. This list is a list of how to transport things into and out of your cell. So this is kind of what it looks like in terms of selective permeability. This is kind of like an image showing you how some of these channels work. And that we're going to talk about that because that's all transport. In this particular case, you've got an aquaporin. So you can see how it's basically a little hole in the membrane that allows safe passage of water to basically freely flow back and forth into the cell or out of the cell, depending on which direction it wants to go. Uncharged small molecules like oxygen or CO2 can freely pass back and forth. Typically, macromolecules, large proteins and things like that, are too big to get through the membrane. So you got to have a different strategy for that. And charged molecules cannot pass through the same channel that water passes through. Generally speaking, if you want something to come in, you have to build a special channel for it. So water has its own channel. Sodium, which is a positive ion, has its own channel. Potassium has its own channel. Okay, so let's go ahead and start taking a look at how we move things through. So we're going to start off first of all with diffusion. And diffusion is basically the random movement of molecules, not just molecules, but solute molecules. That is to say, the stuff that's dissolved in water. And it's always going from a high concentration to a low concentration. And they keep moving until they're equally distributed. Generally speaking, diffusion is what's called a type of tacit moment. That basically means that there's no energy required. It's a downhill process. So if you think about it, it's kind of like going down a slide, right? If you're at the top of a slide, it's a passive process to go down the slide. All you have to do is just go, and gravity will take you down. That's kind of the way diffusion is. So generally speaking, whenever you take a look at diffusion, what's going to happen is you're going to have essentially this disparity in concentrations. So for instance, on one side, you're going to have a high concentration of molecule X. On the other side, you're going to have a low concentration of molecule X. What's going to happen is diffusion is going to occur. It's going to flow from high to low. And as it flows down from high to low, then this concentration, what's called a concentration gradient, will get a little less steep. So this is fast diffusion. This is slower diffusion until ultimately the two concentrations of molecule X are at the same. This is no diffusion. Think about it. if you have a steep slide, you're going to slide down very fast. That's like the big concentration gradient. If you have kind of a shallowish slide, then you're going to go pretty slow, right? That's kind of like the middle case. If you just basically have a slide that's just flat on the ground, you're not going to go anywhere because there's nothing pulling you down. That's the last case, no diffusion. That's equilibrium. Generally speaking, when molecules are moving, they move either way, either into the cell or out of the cell, doesn't matter. All they're doing is they're always flowing from high to low. And so generally speaking, once you get to equilibrium, you essentially have the same number of cells kind of moving in versus moving out, but you still have molecules moving. So that's kind of what it looks like. So here's the initial setup. So notice what we have over here. So purple is the solute. Water are these little Mickey Mouse ears things. So what you have over here is basically a high concentration of your solute. So what that means is your solute is going to want to go this direction by diffusion, high to low. Water typically will go the opposite direction. So once they start moving, they'll start to move down their concentration gradients back and forth until they come to equilibrium where it's all the same. So concentrations are equal over here. Now, let's compare this with osmosis, right, which is the movement or the diffusion of water molecules. Now, typically speaking, they flow from high to low water concentration. Or you can also think of this as they flow toward salt. Matter of fact, there's a mantra in osmosis to help us keep it all straight. We say salt sucks. So water always chases the high salt concentration. Now your body fluids are what's called isotonic. That basically means same salt. It's balanced. And so as a result, generally speaking, the cells don't change size. That means they don't take on water. Why? Because you have equal exchange or isotonic, same salt, same concentration, equal exchange of water. Now, in a hypotonic solution, so in an isotonic solution, if you want to kind of take a look at it, let's just take a look at it from the perspective of the solutes inside versus outside of a cell. So here's a blood cell, for instance, versus your surroundings. So typically in an isotonic solution, the solute concentration, the salt concentration outside the cell is equal to the solute concentration inside the cell. So they're the same. So there's equal transfers. So there's no net movement of water. In a hypotonic situation, what you have is the solute on the outside is less than the solute on the inside. So the salt is in the inside now. The cell looks salty. So water is going to move toward the inside. And what's going to happen is your cells are going to swell just like a water balloon, and they're going to burst. That's not a good thing. That's called lysis. In a hypertonic solution, this is a situation where the solute concentration on the outside is greater than the solute concentration on the inside. So what's going to happen is the water is going to go out. It's called crenation. So the shells will shrink and they'll dehydrate. So generally speaking, this is the movements of water, either into or out of the cell, depending on what kind of salt concentration you have, either inside or outside of the cell. This movement of water is what's called osmotic pressure. This is what drives osmosis. So remember, water chases salt. Salt sucks. And that's osmosis. So here's what it looks like. So under isotonic conditions, you basically have equal transfer of water going in and out because the saltiness is the same inside and out. Hypotonic solution, you have watery matrix on the outside. It looks salty on the inside. So what's going to happen is water is going to go in. It's going to blow up those cells, and then those cells basically are going to burst. If it's a hypertonic solution, that means it's the salty on the outside. So that basically means the water is going to get sucked out of the cells, and they're going to crenate, and they're going to dehydrate. And so that's not good. Okay. So that is osmosis and diffusion. They oftentimes go together as a package deal. Now we want to take a look at facilitated transport. So facilitated transport is basically the transport of molecules across the membrane, still from higher to lower concentration, so it's passive, but they either need a protein carrier, or some sort of a transporter. Basically, this is diffusion with protein assistance. So it's passive because it's still downhill. You're still going from high to low. And these protein transporters are typically specific. They're specific to their molecules. Okay, so if you want a sodium transporter, you have to create a sodium transporter. So here's kind of what it looks like. So in this particular case, you want to basically move this molecule here. So this protein will bind to this molecule here, and then what it'll do is it'll dump it on the opposite side. Okay, so you're using a protein basically here to do this with. That's facilitated diffusion. Then another type of transport we have is active transport. Now this one's different. Active transport is different than passive. It's a major shift. So all the ones we saw before were passive transport. This one is active transport. Active transport is one that requires energy. And so in active transport, you're still moving the molecules, but now you're going from lower to higher. Notice the shift. So this is an uphill process now. So with active transport, it's like basically pushing a boulder uphill. You have to put some energy into that boulder to get it uphill. And as a result, you need energy to fuel this, and that energy comes to us in the form of ATP. That is our energy molecule that we use. Generally speaking, this is driven by a protein carrier, or called a pump. So you'll probably hear me refer to it as a pump. I likely will not refer to it as a carrier. I'll refer to it as a protein pump, or as like a sodium pump. The poster boy for this is the sodium potassium pump, which is active everywhere, and it's a critical pump. So here's how it works. By the way, the sodium potassium pump is one that sort of underlies your entire nervous system. This is how your nerves basically send electrical impulses. So ultimately what's going to happen is your sodium potassium pump has two different pockets called active sites. So an active site is defined as a three-dimensional pocket that binds a specific molecule called the substrate. That's the molecule that your enzyme works on. So basically what happens is the sodium potassium pump has two different active sites. It has active sites here, pockets for sodium. So what'll happen is it'll grab onto 3 sodium, and it'll transport those 3 sodium to the outside of the cell. It also has pockets for potassium. So it'll grab 2 potassiums and then transport those to the inside of the cell. So what it does then basically is it's going to build a concentration gradient of sodium, increasing concentration on the outside relative to the inside, which is low. And for potassium, it's going to be just the opposite. The concentration gradient of potassium is going to be high on the inside and low on the outside. So then, because of that, you've got concentration gradients. And so if you want to, you can actually allow these things to diffuse with facilitated diffusion down their concentration gradients if you want, whenever you want. So they'll want to diffuse in this direction. So basically what you're doing is these pumps build concentration gradients. That's critical to really understand. Concentration gradients do not come for free, right? just like you didn't get up to the top of the slide for free. You had to climb up the stairs. So wherever you see a concentration gradient, like diffusion or whatever happening, always know that there's a pump out there that had to add energy and create that concentration gradient in the first place. Okay. Well, that's all well and good for molecules that are small. But what happens if we get these big macromolecules? How do we move those around? Well, that's bulk transport. So their focus is to move these very, very large molecules across the membrane. You have a couple of different types. The first one you have is what's called endocytosis. So endocytosis basically is going to be moving molecules, and they're going to be moving molecules into the cell. And the way they do that is by basically invaginating the plasma membrane to create a vesicle. So essentially what happens is it looks something like this. So here's your membrane. It'll start to pucker inward. That's what invagination means. It's kind of like an inward pucker. And as it puckers inward, it continues. These two shoulders will touch, and then eventually it'll get so close together that it'll just pop off into a separate vesicle. That's endocytosis. There's three different types of endocytoses. There's phagocytosis. So phago basically means to eat. Cytosis means the condition of the cell. So this is the condition of cell eating. So endocytosis of pathogens. So you basically engulf a pathogen like bacteria. This is what your white blood cells are doing. And you devour it. In a similar process, you have what's called penocytosis. So this is basically cell drinking. Here you take small sips of fluid and small particles from the outside of the cell. And then the last one you have is receptor-mediated endocytosis, where basically what you have are specific receptors that basically grab onto specific molecules. So for instance, with receptor-mediated endocytosis, you would have something like this. So there'd be a receptor out here like this, and then basically what happened is you grab onto a specific molecule, like you're cherry picking molecules out there, and then you just basically internalize those receptors. So let's take a look at this. So here's phagocytosis. You can see you're basically moving that little food particle inward. These two shoulders will touch, and then you'll get your vesicle inside. Penocytosis, similar process. The only difference is what you're taking in. In receptor mediated, you'll see here's your receptors here. You can see they grabbed onto their specific molecule. In this case, you tend to have like a little protein coat to use as a foundation. And then what you do is you pull on that protein coat, and that will basically create your little vesicle. So here you have this coated vesicle. So that's receptor mediated endocytosis. Now, that's all well and good, but now how do you get stuff out of the cell? Well, that's exocytosis. There's only one of those. And so in exocytosis, in the opposite process, this is essentially where you're going to be moving large things out of the cell. It's like reverse endocytosis. So here's your plasma membrane. Here comes a vesicle. As it moves closer and closer, what's going to happen is it'll touch the membrane. And just like two fusing soap bubbles, they'll merge. And as they merge, that vesicle will dump its contents to the outside world, and then you'll just have your plasma membrane. This is basically how you release anything, how your cells secrete, how your hormones are secreted, how your glands secrete things. And then so for instance, one thing can secrete by exocytosis a particular molecule, and then another cell can have a receptor-mediated endocytosis, a receptor that would just basically grab onto that signaling molecule. This is how your cells talk to each other. This is how they coordinate their activities with each other, is by using molecules to do that. Okay, now let's take a look at each one of the individual systems. The first thing we want to take a look at is the nucleus and the endomembrane system. So the nucleus basically, so we're going to be doing like a little tour of the organelles. So the nucleus essentially is going to basically contain all your DNA, your genetic instructions. So it has everything inside the nucleus that is needed to build you from scratch. From that, you also have what's called the endomembrane system, which is basically a series of organelles that are interconnected with each other. that function in the secretion process. So basically, this is how secretion of something works. We'll take a look at that here in just a second. So the nucleus, what's in it? So what's in the nucleus? What's in the nucleus is chromatin and chromosomes. So chromatin, which is basically what you see most of the time, and chromosomes only when the cell is dividing, is essentially your DNA that's kind of bound up with proteins. And essentially, this is kind of like DNA at work. So generally speaking, what will happen is you have your DNA, and it'll be wound around these little protein drum-like structures called nucleosomes. And that basically is kind of what holds on to your DNA. So it kind of looks like little beads on a string. And this is basically what's called chromatin. That's the structure of chromatin. You pack that down a little tighter to make chromosomes, but ultimately what you have here is genes. So genes are basically just sequences of DNA that encode a protein. That's basically what these are. And so your genes make up your DNA. Your DNA is bound to these proteins in the form of chromatin. The nucleoplasm is the fluid inside of the nucleus. And then, of course, you have the nucleolus, which is a dark region. This is basically an aggregation of ribosomal RNA which is a structural component of ribosomes. So that's what's in your nucleus. It's bound by a membrane called the nuclear envelope. It's actually a double membrane, which means you have an inner membrane space, and an outer membrane. And in order to get things into and out of the nucleus, you have nuclear pores, which basically allows safe passage. So this is kind of what it looks like. So here's your nucleus. This guy right here is your nucleolus. Here you can see your double membrane with a little space in between. And then, of course, your nuclear pores here, allowing safe passage of stuff to come into and out of the nucleus. So you have your nucleolus, your nuclear envelope, and your pore. Now, from the nucleus, the outer membrane it is, you see this evagination. So an invagination is a pucker inward, and evagination is a pucker outward. And so this evagination will occur from the outer portion of the nuclear envelope. And what will happen is it'll sort of fall down in sort of like chef hat's flap. And then it'll start to create this sort of labyrinth as you sort of continue to sort of evaginate these little pieces of membrane. They'll kind of fall flat on each other, and then they'll kind of evaginate and fall flat. And eventually you kind of get this labyrinthine structure that we see in close to the nucleus. And typically it's going to be studded with ribosomes, which are essentially making proteins. This is what we refer to as our rough endoplasmic reticulum. A little further beyond that, the endoplasmic reticulum will evaginate and kind of fall down flap-like. And it'll create this large tubular system called the smooth endoplasmic reticulum, or the smooth ER. So you can see that there's basically a nuclear system here. The nucleus basically gives rise to the ER, both rough and smooth. And they have slightly different roles, and we'll take a look at those here in just a second. The ribosome itself, and we did talk about the ribosomes, basically is made up of RNA. That's ribosomal RNA, which is in the nucleolus, and protein itself. Its job is protein synthesis. So these basically are your protein factories. That's what they're doing. Typically, you're going to find them attached to endoplasmic reticulum, or they're going to be free in the cytoplasm. The free-floating ones oftentimes will kind of cluster together to create what's called polyribosomes. And then typically they'll be making cytoplasmic proteins. So proteins that are basically active in the cytoplasm, some other far-flung region of the cell. And so ultimately, they're going to be made by these ribosomes, and then they'll kind of do their job there. So the endomembrane system, if we take a look at it, it's going to basically be composed of a few parts, right? So nuclear envelope, which gives rise to the endoplasmic reticulum. We just saw that. the Golgi apparatus, the lysosomes, and vesicles. So we'll take a look at these. So you're just compartments or journeys on the way of a single specific process. So here you can see your nucleus, kind of taking a look at the entire endomembrane system itself. And take a look at these here in just a second. So I'm going to go back and forth between this picture and the image itself. So the first piece of the endoplasmic or the endomembrane system is the rough ER. Typically, it's studded with ribosomes because it is making proteins. The smooth ER, which is also part of it, which is less common. So here's our smooth ER down here. Basically, the smooth ER doesn't have ribosomes, and so its job is to synthesize lipids. It also does detoxification, and it also does storage of calcium. And so in different cells, it has different functions. Okay, so that's an important one. And then we have the Golgi apparatus, which is this guy right here. This kind of big, looks like a big flap, a stack of pancakes. And so the Golgi apparatus's job is simply to take the proteins that it gets from the rough ER, and it modifies those, and it packages them, and it ships them. Okay, so that's basically the process of secretion, but let me kind of round out the other characters, and then what I want to do is I want to put it all together in the secretory process. So how you get from, say, for instance, the rough ER to the Golgi is by use of a transport vesicle. A transport vesicle simply is a little membranous sac that basically has protein in it. And either that protein is crude or it's ready to be secreted. That depends. Okay. Now, a lysosome, it looks very similar to a vesicle. It's made by the Golgi in the same way, only it's basically differs by what's inside of it. What it has inside of it is what's called hydrolytic enzymes. These basically are digestive enzymes. They break down the macromolecules. So these digestive enzymes will break everything down. So a single cell, basically a lysosome, is kind of like the stomach. It'll basically devour and divide everything up. So when a white blood cell engulfs a bacterium, it fuses the lysosomes to that bacteria to chop it all up and to break it all down. So what about secretion, though? So let's kind of put it all together. So here's how it works. Imagine you have a protein hormone that needs to be secreted to the outside world and communicated. What happens is the ribosomes will start to make the protein in the ER, the rough ER. That protein will fold up, and then what's going to happen is it'll pinch off. It'll kind of bulge outward from the ER with this crude, unfinished product of a protein inside this vesicle. Once it pinches off, then you have a transport vesicle, which has crude protein in it. It's not ready yet. What it does then is it'll come and it'll dock to the cis face, what's called the cis face, or the same side face of the Golgi. And when it does, it'll dump the proteins into the Golgi, which will then be modified by the Golgi, and it'll advance from one stage to the next until it becomes perfected. It'll be sorted out into a special vesicle that will then start to fill up with now the finished protein. And at this particular point, it'll pinch off of the Golgi, and it'll become now what's called a secretory vesicle. And this basically has the finished protein. This is ready to work. So then the secretory vesicle will go to the membrane, fuse with it, and like two soap bubbles fusing together, it'll fuse with it and it'll dump its contents to the outside world. That's secretion. And then that'll go off to wherever it needs to go to signal its communications to wherever in your body that hormone needs to communicate to. Okay. So that's the secretory pathway, and that's basically what ties the endomembrane system together, is that secretory pathway. So now let's go ahead and take a look at a couple of other things like cytoskeletal and cell movement, cell junctions. Now, the reason why we're talking about endomembrane system and cytoskeleton is because if I were to ask you, like, what are some of the features that really sets eukaryotes apart from prokaryotes, these are the two that would do it: endomembrane system and the cytoskeleton. Okay, so let's go ahead and take a look at the cytoskeleton. The cytoskeleton basically is composed of protein fibers, and essentially its job is it's pretty basic. Its job is cell shape. It provides for anchorage and also for movement. So basically, the cytoskeleton is both for support and for dynamic movement. It does both. And so when we take a look at the cytoskeleton, there's three major fibers to it. So there's the largest ones, microtubules, which are the ones we study a lot. There's ones that are kind of middle-sized, intermediate filaments, which tends not to be studied as much. And then, of course, actin filaments, which are the smallest of the three. It's like the little brother. So let's go ahead and start off, first of all, with the microtubules, and then we'll kind of work our way through. Basically, microtubules, the way that you assemble a microtubule fiber is through an organelle called a centrosome. And so what this does is basically microtubules are like tracks that basically organelles will move along. And so when you have a microtubule, they typically form as like a long cylinder. And you have this organelle that needs to move from point A to point B. And it moves basically by what's called a motor protein, which has a walking domain, just like our legs. And it has a cargo binding domain. In this case, it's binding the vesicle. Now, with ATP, that's your energy, right? You're going to fuel this process. So it's going to take a step, and the protein is literally going to move down the microtubule. So what you see are organelles moving down the microtubule. They're also important in cell division because they form what's called the spindle, which moves the chromosomes in the act of cell division. Okay, so microtubules are very important for locomotion in the cell. Now, then actin filaments are made of a protein called actin. They're typically be long and thin, and they're usually in movement. So, for instance, this is a main fiber in muscles. So it's a very common one that we see. And then intermediate filaments they have various functions, but most of the time they act as a scaffold for the cell, kind of like a framework. This sort of keeps the cell's shape intact. Okay, so what are some structures that make use of these? Well, first of all, when we take a look at movement, we have cilia and flagella. So cilia are basically short little fibers like this that basically wave back and forth. Flagella are basically the same structure, only they're very long, and they kind of whip back and forth. They're both made of microtubules. They're both associated with movement, either at the cellular level or subcellular level. Cilia are important because they line our respiratory tract. So in your respiratory tract, what happens is you've got a line of cells called pseudostratified ciliated columnar cells. And so what happens is these guys will basically have these little cilia on them. And this is what's in your respiratory tract, like in your trachea, like little hairs. And they'll also produce mucus. So when they produce mucus, you kind of get this sort of thick, mucousy layer with these little cilia hairs. Now what happens is when you breathe in dust particles, it gets stuck in the mucus, and the cilia will sweep by waving back and forth like a kelp forest in an ocean current. And they'll basically move those dust particles up into your respiratory tract where you can cough it out. Okay, that's what cilia does. Flagella, basically, we only see it in sperm, right? So when you take a look at it, this is what cilia looks like. You see like these little hairs, and this is what a flagellum looks like. But basically, they all have this same structure. It's what's called the 9+2 structure. And it kind of is this long thread that kind of whips back and forth for locomotion. Okay, so let's go ahead and take a look then at the extracellular matrix. So the extracellular matrix is basically the outside of the cell. It's a matrix. Of proteins that the cell makes outside of itself. So it secretes it. So here's your little cell. What'll happen is it'll actually secrete these different molecules outside of it, and it'll create this little extracellular matrix. And there's a lot of different things in there. So there'll be some polysaccharides out there, some proteins out there. One such protein will be collagen. So collagen is tough and structural. So this gives a little bit of structure to the outside of the cell. Elastin, it'll bump that out there. This is flexible and bendable, so that'll give you some flexibility on the outside. Another thing we see is what's called fibronectin. This is kind of like an adhesive protein. And so what happens is it binds to a protein called integrin, which is actually inserted into the membrane. And on the other side is actually tethered to your cytoskeleton. And so this kind of helps bind or anchor the cell into a stable place. And that's kind of what your extracellular matrix does a lot, is it creates an environment for your cell to be able to be functional in. It also creates stability in your cell. So this is kind of what it looks like. So here you can see this kind of polysaccharide proteoglycan mixture that's kind of out there. It's kind of a semi-gelatinous goo on the outside of the cell. The cell can pump in as much collagen as it wants. This gives you tough structural, like rebar in cement. You can also add some elastin in there if you want a little bit more flexibility in what's around you. And for anchorage, you have your integrin. So here's your integrin protein. You can see it's basically inserted into the membrane. And on the inside of the cell, it's basically attached to actin filaments. So this piece right here, this actin, there's a region of actin just underneath the cell that we refer to as cortical actin. And it's pretty solid. It's like a solid matrix. So basically you're tethering your integrin to your solid matrix cytoskeleton on the inside. On the outside, it's attached to fibronectin, which is this little green thing, which itself is then attached to collagen. So basically what your cell is doing is anchoring itself to this tough collagenous structure, and then anchoring itself by way of its cytoskeleton to its extracellular matrix. So that stabilizes the cell itself and allows it to be able to be stable. Okay. What about battening these cells together, putting them together, binding cells together? That's cell junctions. There's three main ways to basically fasten cells together to create tissues. So we use these to create tissues. So the first one we have is what's called adhesion junctions. So they attach cytoskeletons to adjacent cells. We have tight junctions, which are kind of fasteners, like buttons, rivets, if you will. They produce a pretty strong barrier. And then we have gap junctions, which typically are channel-like structures that allow free communication between cells. So here's kind of what they look like. So here's our adhesion junction. Notice how you have proteins that are associated with the membrane. They're embedded in the membrane. They kind of reach across each other and they kind of lock on. So these two receptors will actually hitch to each other, just like two train cars hitching to each other. And then they kind of anchor themselves to this actin cytoskeletal framework. So you're doing a very similar sort of a strategy to what we just saw with integrin and fibronectin. Notice also you have a little bit of space here. And this can allow some communication between cells. Now in tight junctions, you're going to notice that these guys are really tight. So they're just basically fastened in there. There's essentially no space. So you're not using this one to communicate. You just want a really tight, solid barrier, impenetrable barrier on this one. Then, of course, we have the gap junctions, which are essentially channels between two cells. So whatever happens in the cytoplasm of cell number 1 will basically be communicated in the cytoplasm of cell number 2. So essentially, if a message comes into cell number 1, it'll flow right through, and cell number 2 will hear the same message. So this is basically for max communication. So what one cell hears, the other cell hears almost immediately. Okay, now let's go ahead and do a little bit of a roundup of metabolism, just because we need energy to fuel all of this. So when we take a look at metabolism, this is basically all the chemical reactions that occur in the body. And so they're broken down into metabolic pathways and different types of metabolic pathways. They generally will start with some sort of a reactant and end with a product. So for instance, you'll have a reactant, the reaction will go forward, and then you'll have your product. And oftentimes you can have multiple steps. So you can have many steps associated with that on your way to your final product. And generally speaking, when we take a look at enzymes that sort of move or work on these enzymes or these substrates, we refer to these molecules that they're working on as substrates, and they're converted into different products. So these enzymes basically just kind of convert them into different products. This is kind of an example of a metabolic pathway. So this is kind of a multi-step one. So here we have substrate A. Substrate A is going to be bound by enzyme 1. It's going to be changed into substrate B. Substrate B will then be bound by enzyme 2, which enzyme 2 will manipulate it and turn it into substrate C. Substrate C will then be bound by enzyme 3, and then will be turned into the final product, D. Typically speaking, we refer to all of these as intermediates. Oftentimes things we don't even notice or we don't even see. Now, generally speaking, whenever you're controlling something like this, we have a type of regulation called feedback inhibition, which is essentially where the end product, D, will shut down its own production or stimulate it, right? So for instance, the accumulation of the product will come back here and either positively affect it or negatively affect it. It'll either upregulate it, really turn it on, or it will shut it down. And so we see a lot of these actually in physiology. So since we talked a little bit about enzymes, let's kind of take a little bit more of a closer look at enzymes. So enzymes basically are what we refer to as biological catalysts. And a catalyst has a two-part definition. You can't be a catalyst until you satisfy both. First of all, you have to speed up a reaction. And secondly, you cannot be consumed by the reaction. Those two are true. So as a catalyst, enzymes will speed up the reaction. They'll basically take their substrate, they'll do some sort of a chemical reaction on it, break it down, for instance, like a lipid, for instance, will be broken down into smaller and smaller lipids by an enzyme known as lipase. And so different types of enzymes will work on different structures, but it's breaking things down. Notice oftentimes enzymes will have the suffix ase. So this oftentimes means you're working with an enzyme. So when an enzyme binds to its substrate, it binds it with that three-dimensional pocket called an active site. We saw that already with our sodium-potassium pump. This is where it binds. And then ultimately the enzyme can just return and do its job again. So here's kind of what it looks like. So this is a degradation enzyme. So first of all, what's going to happen is this enzyme has an active site specifically designed to fit its substrate, the molecule that it works on. So it's going to bind to the molecule. So now you can see that the next stage is this sort of enzyme bound to its molecule. And then what it's going to do is it's going to do the chemistry on this. So the chemistry on this one is to basically cleave these guys apart. And as soon as it does that, then these two molecules are different molecules. And the enzyme will release the products, that's the products there, and then it'll basically be returned back to its native form so that it can go and pick up another substrate and work on some more. So enzymes have the ability to just basically go back and forth and just kind of keep working on their substrate over and over and over again. In the case of an enzyme that synthesizes something, it's very similar. In this case, it's got an active site, a two-part active site that's designed to bind to these two little substrates, in this case, the two different molecules. When it gets them into the active site, it links them together with some sort of a bond, and then it will release the final product, which in this case is the whole molecule. Notice again, though, however, the enzyme is returned to its original state, making it a true catalyst. Okay, so one of the important things about enzymes is that when an enzyme is activating or catalyzing a reaction, the reaction it catalyzes doesn't just naturally go forward. And the reason is because there's an energy barrier in between the reactants and the products called the activation energy. And that's kind of what's here. So here you can see you've got a reactant that wants to go to product. And one would think it should be an easy trip to just go straight down to a lower energy product, right? Because that'd be kind of like a downhill process that's energetically favorable. But in reality, what happens is there's this energy barrier between the reactants and the products that the reactants have to get over. Generally speaking, the reactants can't get over that. And so it's kind of like you going down the slide. You can't go down the slide unless you overcome the energy barrier of climbing up the ladder first. And that's what it's like. So what enzymes tend to do is they tend to basically catalyze reactions by lowering the activation energy. And then what happens is once they lower the activation energy, which here you can see a catalyzed activation energy. So an enzyme has come in here and lowered that. That makes it low enough for the reactant to just basically skip over that little barrier and go straight to the product. Now, some enzymes have some help. They have either their enzymes or they have coenzymes or cofactors. These are just other things, vitamins and minerals and things like that, that are necessary to help the enzyme process its substrate. So the reason why you have activation energy and why that's good is because what this does is it basically means that all your reactions are kind of by default in an off state, giving your cells masterful control over when, how, and where these reactions will turn on. All they have to do is make an enzyme to lower that activation energy, and then it'll effectively turn on the reaction. So what it does is it basically gives you control. So you can determine and dictate how this reaction moves forward and when. So then, let's take a look at mitochondria, which is a little bit of a cellular, since we're talking about energy, right? Because mitochondria essentially is the organelle responsible for creating energy. So what it does is it takes chemical energy and turns it into our rechargeable battery, ATP. Think about ATP as your cell phone battery. If you want a functional cell phone, you basically have to plug it into the wall. You have to recharge it. Same thing's true for ATP. This is what cellular respiration is all about. That's what's happening in mitochondria. It basically uses oxygen, produces CO2, but in the process, it's making ATP. This is happening in your mitochondria, which has both inner and outer membranes, and the inner membrane is folded up into little folds called cristae. We'll see this here in just a second. They also have their own DNA, right, which we already talked about. That was kind of a holdover from the endosymbiosis idea. So ultimately what the reaction is doing is it's basically taking a molecule of ADP, which is adenosine diphosphate plus a phosphate, and it's turning it into ATP, which has a lot of energy in it. And then when you go to use the ATP, you do what's called a hydrolysis. You liberate that, and you generate energy. So a little bit of energy, some ADP, some phosphate will give you ATP. That's your rechargeable battery. You can go the opposite direction when you need that energy by just basically plugging it in and turning on the cell phone, so to speak. Okay, so that's kind of what a mitochondria looks like. So here you can see the outer membrane, the inner membrane, but the inner membrane is folded up into these little folds. That is basically to increase surface area. Why? Because all of your cell respiration enzymes are embedded in these inner cristae folds. And then, of course, here you have the fluid region inside is the matrix. And so the regions of the mitochondria are important because you have different types of cell respiration activities happening in these different regions. This is a transmission electron micrograph of a mitochondria. Probably one of the most distinct organelles in the cell. Nothing looks quite like a kidney bean with tiger stripes. So this is basically our ATP ADP cycle. This is what I just kind of cheated on a little bit. I just kind of went there, right? So basically, you have ATP, which has lots of energy in it. When you hydrolyze it, it goes to ADP and phosphate, but it releases energy that we use for work, for some sort of work. Then we want to recharge it. It goes through cell respiration. The energy that we eat is then packed into ADP and phosphate, and it basically goes right back to ATP. So the energy in your ATP is coming from the food that you eat. So when you take a look at the process of cell respiration, it starts off basically with glucose, which is a highly energetic carbohydrate molecule made in photosynthesis. And there's three main pathways. The first one we go through is glycolysis. And then from glycolysis, we go through citric acid cycle, or Krebs cycle, because it was discovered by Hans Krebs. And then we get to the electron transport chain, which is really where we are able to maximize our energy production. So these three together basically allow for glucose to generate a lot of ATP, or energy, for us. And so here's kind of what it looks like. So you take in a glucose molecule. That's what's basically happening. Insulin, by the way, is a hormone that's designed to stimulate cells to take up glucose. Glycolysis will happen in the cytoplasm. And so you'll go from glucose to a molecule called pyruvate, and you'll generate a little bit of ATP. And then it's going to be moved into the mitochondria, where you go through a preparation reaction. And then you'll start to generate these little electron shuttle type proteins called NAD and FADH2. And then from the preparation reaction, you'll move that molecule into the citric acid cycle, which will generate more ATP and more of these little electron shuttles. And then from there, these electron shuttles will dump their electrons, these electron carriers, into the electron transport chain, which will make a ton of ATP. So basically, you'll make between 36 and 38 ATP per glucose molecule with this particular process. That's a lot of energy. So ultimately, when you take a look at glycolysis, it's going to take a glucose molecule and it's going to break it into 2 3-carbon molecules that ultimately will become pyruvates. This is important. Why? Because from here on out, this doubles everything. So you doubled your productivity. Generally speaking, it's happening in the cytoplasm of every cell. And everybody does it, whether you use oxygen or not, because if you're anaerobic, it doesn't matter, because you can still do glycolysis. It doesn't require oxygen. This is not the part that requires oxygen. And what you get out of that is NADP or NADH, which is an electron carrier. It's just basically like a taxi. It picks up electrons and it goes and takes them to electron transport chain, and also 2 ATP molecules. From here, then, you go into, this is only true when oxygen is available, you go into the preparation phase. And so your pyruvate will enter the preparation phase. And then once it moves into the preparation phase, it'll prepare the molecule for entrance to the citric acid cycle. If oxygen isn't available, then you move into a fermentation pathway, which is anaerobic respiration. So let's imagine we have oxygen. We just gone through the preparation phase. Now we're moving our molecule into the citric acid cycle. This is a cyclic series of probably about 10 to 12 different enzymatic reactions. They occur in the matrix. That's the fluidy centerpiece of the mitochondria. And it continues to break down the glucose. So each pyruvate will enter. It'll enter as a specific molecule called acetyl-CoA. That's the product of the preparation phase. What it produces is more electron taxis, more ATP, and more CO2. And then once it gets done with this, then ultimately it creates a lot of these electron taxis, NADH, FADH2. And then it's going to basically move these electron taxis to the electron transport chain. So NADH from glycolysis and the citric acid cycle will deliver their electrons, just like a good taxi carrier, and it'll move them into the electron transport chain, where proteins are embedded in the cristae. That's the folds of the inner membrane. So in the inner membrane, basically looks something like this. So what happens is NADH will come in, it'll drop its electrons off, and then what'll happen is this protein here, which is a carrier, will actually, it's a pump. It'll take a hydrogen ion and pump it into the intermembrane space. And then it'll pass those electrons on to the next pump, which will take another hydrogen ion and pump it into the intermembrane space. And then it'll pass its electrons on. And it'll take another hydrogen ion and pump it into the intermembrane space. And then that final electrons will basically be picked up by oxygen and hydrogen, and it'll leave as water. This is the reason why you need oxygen for cellular respirations, because oxygen is what's called the terminal electron acceptor. If oxygen doesn't pick up those last electrons, then it backs the system up, electron transport shuts down, and we're not able to make ATP. Then with this energy that we have, now we have a concentration gradient. High hydrogen ion on the outside, and low here. So what happens is we use this energy in the concentration gradient to transfer that to making ATP. So as this hydrogen ion flows from high to low, it releases energy, and we use that energy to basically make ATP. Okay. So this is aerobic, right? So your oxygen receives the electrons, it combines those hydrogens, it becomes water. That's why you breathe out water vapor as a byproduct. And then what will happen is that that ATP synthase will use that energy in that hydrogen concentration gradient to make between 36 and 38 ATP for one glucose molecule. So what happens if we don't have oxygen? Well, then we switch into what's called anaerobic. It doesn't require oxygen, fermentation. So in this particular case of fermentation, after glycolysis, remember, you still have pyruvate and you still have NADH. You got to deal with these. So what happens in fermentation is, in us, we actually just basically dump the electrons onto the pyruvate. And when that happens, it turns into lactate or lactic acid. This is what causes that burn in your muscles when you're working out and you haven't gotten your oxygen levels up just yet. Okay. So that is the end of chapter 3. I will probably do chapter 4 either later today, or you could expect chapter 4 to load up early next week.